Good evening, Pratik. Dr. Sarin here. So,
Hello, good evening.
good evening. So, thank you for joining. We will go live at 7 o'clock. So, at 7 p.m., I will introduce you just for
Okay,
a minute and then you can start your lecture. Thank you.
okay, sure. Yes.
Hello, good evening, everyone. So we are starting with the fifth lecture of this course, and today it's a holiday, public holiday in Mumbai, but we are continuing with the course. So today I want to introduce Dr. Pratik Chandrani. He is a scientist. He's a principal investigator and an assistant professor in ACTREC Tata Memorial Center, and he has his own group and with PhD students. And if I have to describe him, he's basically a computational biologist and a data scientist, and someone who has worked very closely both with biologists, cancer biologists, as well as clinicians, as well as pathologists, and also participates in molecular tumor board. So he just doesn't work only sitting on his system. He interacts with real human being, and he knows what queries come. So I have requested him that he keeps it a little simple, because all of us know that bioinformatic analysis is very important, but we have our own concepts, and some of those are misconceptions. Some terms we don't really understand. So he has told me that he has kept it, addressing biologists and clinicians. So please start, Dr. Pratik.
Thank you so much, Dr. Rajesh Sharan, for nice introduction and this kind invitation. It is my pleasure to present today primer to computational biology of NGS, and this content has been specifically tailored for biologists and clinicians. But still, if you face any difficulties, feel free to ask the questions in the last 15 minutes of question answer session, as well as please feel free to contact me via email or any other modality afterwards if you need. So, very good evening, everyone. I'm Pratik Chandrani. I'm a scientist at ACTREC Tata Memorial Center, and my lab basically focuses on cancer genomics and other data-driven approaches for identification of novel therapeutic targets and novel diagnostic and prognostic methodologies for cancer. So today, before we jump into what we need to do in computational biology of genomics, first let us see what we can actually achieve by doing this hard exercise. Because few slides down the presentation, you will see that we'll be talking about lot many complicated concepts. And first of all, we need to have enough of the reason to know what we are going to achieve by doing this hard exercise. So in this slide, what I'm showing is a exercise from my lab in which we profiled 1,000 different tumor samples of Indian origin by using comprehensive genomic characterization. And this is a humongous exercise of so many people. I'm just merely making a presentation here. But when we started asking these all questions of cancer genome in India, we realized that we don't have enough of the data, and Indian cancer genome has not been well characterized. So we formulated a team of fantastic, motivated biologists, medical oncologist, pathologist, and our NGS team expert. And combined together, what we have is our first report, which is in communication as of now, in which we were able to profile tumors across 20 different cancer types across the body. And through this, we were able to identify various different immunotherapeutic biomarkers like tumor mutation burden, microsatellite instability, PDL1 expression, high PDL1 expression. And you can see that all three of them are complementary to each other. The commonality between three is very much minimal, as you can see here, but they are all unique and individual contributors for identification of immunotherapy suitable patients. Hence, all three are needs to be put into practice for regular clinical care. Furthermore, what we see interestingly is huge repertoire of therapeutic biomarkers. You can see here row-wise all these cancer types are there, and column-wise you can see various different genes which have been well established in clinics or they have some novel attrition. So you can see here color-coded bar chart. This dark pink color indicates that whatever gene mutation has been observed in Indian patient has some drug candidate available as per the US FDA approval, as well as there is a drug candidate available as per the Indian DCGI approval, and hence it can be directly given to the patient. Now one can quickly see that out of all the sequencing effort that we did, very tiny fraction of patients are currently covered through the DCGI approval, and there is a huge potential for further development. So we break it down into various different parts as well, the type of development required. So one can see this pink color. This pink color indicates that there is a biomarker present. This biomarker is, as per the word FDA agency, it is approved for therapeutics, but it is not yet approved by Indian DCGI agency. Hence, some set of phase 2, phase 3 clinical trials should be focused on all of these cancer types and the biomarkers of their corresponding counterparts to bring this targeted therapy to India as soon as possible. The light pink color that we get to see indicates that there is some drug approval available at the international level in the same gene, but not for the same mutation that we have observed in the Indian patient. And some form of preclinical work is required, but we don't need very exhaustive preclinical work. So here, somewhat, preclinical work, a quick validation in cell line would be good enough to proceed to the phase 2, phase 3 clinical trial. And the largest part we get to see is all gray bar chart. This gray bar chart indicates that there is no drug approval at the worldwide level, including the Indian DCGI. So this is where a more of next phase of biological research is supposed to be focused, and Indian biological laboratories should pay attention to this kind of cancer types and their respective biomarkers. Go to the cell lines and use animal models and validate that these are driver alterations, and also validate that they are sensitive to the small molecule inhibitors so that a prospective clinical trial can be set up. So this is the kind of output that we get to see. And here in this one single slide, I'm trying to summarize that how just one single experiment of 4,000 genome in India is capable of transforming cancer care drastically. We have something which can be applied immediately. We have something which can be quickly applied by performing directly phase 2, phase 3 trials. We have something which requires a little bit of preclinical work and then phase 2, phase 3 trials. And we have something which requires extensive biological work and then it can be brought to clinics. So if we want to find out something like this, and if we want to bring this to clinical practice, what we need to do in today's talk, we'll be talking about genomics related to that. So first of all, you can quickly recall the genomics library preparation and other wet lab workflows were greatly covered by Dr. Ramsir Shetty in lecture number 4. If you need, you can go to YouTube link and find out the details. But to quickly summarize, basically we get started with DNA or RNA. We in molecular biology, we are not so familiar with RNA. We are not so comfortable working with RNA, as majority of the molecular biology tools have been developed for DNA. So we can convert this RNA into something called complementary DNA by using reverse transcriptome. Once we convert it to cDNA, then everything becomes logically very similar. So we have this DNA or cDNA as a starting material. We chop them up into smaller fragments. These fragments are then ligated with adapters at both the ends. These adapters are universal adapters so that our sequencing machinery can bind to this adapter and perform the sequencing. We load this into the sequencing chip where they form clusters, and these clusters are then monitored by very high resolution camera in the sequencer machine. And in this cluster, when every base pair is added in this fragment, it is captured by the camera and then later on decoded into NGS read. So this is a bird's eye view, you can say, of NGS sequencing. I'm not trying to misguide you in the way that this is a very simplified process. No, it is not. Here I have oversimplified it. It is very complex process and it requires a lot of extensive hands-on training. But once we do this, once we have the sequencing data, then what do we do? So usually sequencing data comes in a pair of read. So in each of the fragment, we have sequencing primer ligating on this side, and then it will perform sequencing in this way. So this will called forward read. Then we'll remove all the ligated molecules, and then we'll use another set of primers which will bind on the opposite side of the adapter, and then it will do sequencing from the opposite side. So we call it reverse read. So on every DNA fragment, we have forward read and we have reverse read. This is typically of 150 base pair in length, and our fragment is typically of 400 base pair in length. On every 400 base pair, we have 150, 150, total 300 base pair covered, and then we have 100 base pair gap in between these two. So this is a most typical sequencing scenario using Illumina sequencing platform. However, there are some other varieties of sequencing platforms as well, which does not follow this paired and sequencing protocol. But those are not as widely in practice as Illumina. So for this today's talk, we'll restrict everything to Illumina. So once we get the sequencing read, the first thing we need to do is align them to the reference genome. Now we know that human genome has been available since 2001, and we have several improved versions of human reference genome. Our current standard is GRCH 38 version, which is global standard for cancer genomics analysis as well as for any sort of human genomics analysis. So we take that genome sequence from NCBI. This is the genome sequence reference. We take our NGS data, align it to the reference genome so that we can find out where they are mapping. So each of these read pair you can see is mapping at various different locations in the genome. And after this alignment, we get to see what kind of alterations are there. Say, for example, in the reference genome, we have A base pair, but in the sequencing data, we see C base pair at various different locations. So we can quickly count it. In this particular location, we have total 18 reads. So our coverage is 18x, out of which 7 times we have A, where nothing is written, it is the reference base pair, and when there is alteration, altered base pair will be written over there. So C has been counted here at 11. So 11 out of 18 is equivalent of 0.6, which is allelic ratio or allelic fraction. Now one can quickly recall from the basics of cancer, basics of genetics class from masters or bachelors. Human genome is a diploid genome, so we have two copies of the genome. And if you are to do say 100x sequencing, 50x will be representing one copy and 50x will be representing another copy. So whatever x coverage you have, you can always figure out what is the proportion of each of the copy of the DNA from the cell. So here, if it is 18x, a typical heterozygous mutation would show 9 times mutation and 9 times wild type. Here we are getting very similar number, so one can easily call that this is a heterozygous mutation. If this would have been a homozygous mutation, you would have observed C 18 times out of total 18x coverage. So that's how we find out allelic fraction and we interpret whether it is a heterozygous mutation or a homozygous mutation. Similarly, one can easily find out if there is any insertion or deletion, in short called indels. So one can see here that there is a small piece of DNA missing in our sequencing data, but it is present in the reference genome. So there is a deletion event happening here, a small deletion, and that can also counted in the very similar manner to single nucleotide variants, SNV. Then other type of variants are copy number alteration, in which either there is a whole bunch of DNA region deleted, which is called homozygous deletion, or there is less than half of the coverage compared to the average coverage of the sequencing. So we call it hemizygous deletion, single copy deletion. If we have more reads than the average, then we can say there is an amplification or copy number gain in this particular location. Furthermore, if we have this read pair in which forward read is mapping to chromosome 1 and reverse read is mapping to chromosome 5, or for that matter, these two reads are mapping to any two distinct region of the genome, not at the 4 base pair apart, but longer than the 400 base pair apart, then we know that there is some kind of gene fusion happening in the DNA. So for the sake of simplicity, let us say chromosome 1 and 5 are fused in the patient sample. If that is the case, then only we'll get to see a read pair in which half of the read is mapping to chromosome 4 and half of the read chromosome 1 and half of the read is mapping to chromosome 5. So this is how we find out a translocation or a genomic fusion by using NGS. And then if we have NGS reads which are not mapping to any of the human reference genome, but if they are mapping to the non-reference genome, say for example, in a cervical cancer case, we might have HPV, human papilloma virus DNA present as well. So whenever we do whole genome sequencing, we'll always find HPV in almost all of the cervical cancer patients. So in our NGS data set, whatever millions of reads we identify, a few fraction of them, a few thousands of those reads will not be mapping to human genome. They will be mapping to HPV genome, and that's how we can find out non-human, pathogenic infections from a human sample. So this is, in short, conceptual overview of computational analysis of NGS dataset. But now let us get into more details because we don't want to just see the overview. We want to put this in practice. So to put this in practice, we often do one thing in cancer genomics is that we focus on various genomic region and we don't target the whole of the genome. Our genome is about 3.1 billion base pairs, and that is just too much for the sequencing, and not entire genome is as informative is few segments of the genome. So here I'm showing you a graphical view of a human genome, all the chromosomes, and the red bar indicates the location where a therapeutically relevant mutation has been observed in the genome. So in summary, in our in-house clinome-based database, which I developed when I was a postdoc in that laboratory at ACTREC, we were able to catalog about 3,000 different mutations across the genome, which were attributable to roughly about 200 different small molecule inhibitors and various other therapeutic agents. So, in clinical genomics, we often target the small region. So, what are different targets and how do we do? Let us quickly go through that. So, one is whole genome, in which we sequence everything, and then we have whole exome, in which we sequence all of the coding DNA. Then we have whole transcriptome, in which we sequence all of the transcripts by taking, by converting them to cDNA. And then we have targeted and amplicon or panel sequencing, in which we target very small number of biomarkers, typically 10 genes, 12 genes, 52 genes, 71 genes, and 500 genes are most commonly available for various different cancer types. So they are also much more cost effective and faster compared to other modalities of the sequencing. So once we get the sequencing, we also need to factor in which platform we are analyzing with. So in today's talk, we'll be focusing majority on Illumina-based platform, which are most commonly present in various different diagnostic practice as well as in research practice. But there are some other platforms as well, like iTorrent, which does not do paired sequencing. It provides only forward read sequence. And then we have third generation sequences as well, but they are not yet in the clinical practice and mostly suitable for research practices only. So now to get into some more details of how exactly we perform the sequencing analysis, we need to understand various different file formats. And this becomes very critical. In a couple of slides down the line, you will see that there are several different steps of the analysis, and in every different step, if we keep producing files as per individual liking, then this analysis cannot be communicated globally. This analysis cannot be conveyed globally in an easy manner. So there are consortiums which defines file format at the global level, and all of the programmers, all of the computational biologists like us, whenever we develop a program, we strictly follow these file formats. And even if you are not from a bioinformatics background, if you are from clinics or basic biology background, still it is essential for you to know the basics of this file format so that when you are dealing with it, you accidentally don't modify it in a way that it will break the file. So the first file that we deal with is called FASTQ. This is the file that we obtain from NGS platform. This is called raw sequencing data or a raw sequencing file. This FASTQ file has a four-line structure. First line is always sequence ID. The sequence ID is broken down in this table. There are various different numbers of this kind, which indicates instrument number, flow cell length, and other details related to the machine. Now, this is something which you don't need to manually open and read. This is essential only for bioinformaticians so that they can read this information when they are writing the program appropriately. So this is an example of sequence ID. If you actually open FASTQ file, this is how sequence ID will look like. And if you are interested in more details, feel free to consult this table afterward. Then the second line is the sequence itself. This is where the nucleotide sequence is stored. Here you will get to see only one sequence read only. In the single file, we don't club forward and reverse read together. So always in the Lumina setting, we will have one read file, one FASTQ file called forward file, and then another called reverse file. Or sometimes we refer to this as 1 and 2. So for patient one underscore one dot FASTQ, patient one underscore two dot FASTQ. So these are the paired and rich stored in two different files of particle format. So this is that nucleotide sequence itself. Then the third line is always starting with this plus symbol, and then optionally it can have the same sequence ID at the first line, or it can be just a plus symbol. And then the fourth line is very critical. It has Fred quality score. Now, Fred is something very similar to Sanger sequencing, which most of the biologists and clinicians would be more familiar with. In Sanger sequencing, you get to see four color spectra. The spectra is usually having a fixed height from 0 to 40, and that height indicates the quality of the nucleotide sequence at that particular location. So the same Phred quality score is employed here in the NGS platform as well, and basically this is a logarithmic scale. So if there is a 40 quality score, that means 1 in 10,000 nucleotides sequenced in a particular setup is having a likelihood to be a false positive. 30 means 1 in 1000, and so in the logarithmic scale. And these are the ASCII symbols of each of the value. So computationally, we can't write these numbers because if we have, say, 150 nucleotides here, and if we have two-digit numbers, 40, 40, 40, sometimes it can be a single-digit number. Quality score is 1, 2, or 5. So it will be difficult to find out which number is associated with which of the nucleotide. So in computational language, we have something called ASCII table, which is ASCII symbol. These are single character symbol, and each symbol represents one particular number as per the table index. So these are fixed and these are universally, applied across all the computers. So this is easier to read for a computer and interpret. So this is how a PathQ file is produced. Now this PathQ file is further processed in the computational analysis pipeline, and during the processing, we produce a file called SAM, sequence alignment and map format. And after getting the alignment to the reference genome, we also convert it and compress it in a more compressed format called BAM, which is a binary alignment map format. It is nothing but a binary file. This SAM is a text file, a human readable file, and this BAM is not human readable file, only computers can read it. But that will compress the size as well, so that helps us in saving some computational storage space. In this SAM or BAM file, we have multiple lines starting with @ symbol. This is called header line. Header lines contains information relevant for various different experiments. Say, for example, here this is indicating that we have done alignment against NCBI version 37 of the reference genome, and it is not using 38, the most latest one. So based on this SAM file or BAM file header, one get to know whether it has been produced against version number A or B of the reference genome. Similarly, we are also indicating that this is Illumina-based NGS data, and this is the sample ID, so that we can later on figure out whether this sample belongs to some patient A, B, C, or whatsoever. And then after the header lines are over, we see one line entry for each of the read, which is mapped to the reference genome. Again, this has very much complex data, and I have broken it down into the simplified table. If you are interested from programmatic point of view, please feel free to consult it further, and also let me know if you need any advanced help. But for the sake of simplicity, let us look at the parameters which are most critical for the wider audience, including biologists, the clinicians, and practitioners. So in this Sambam file, what we get to see is chromosome position where it is aligned. So on chromosome 1, this particular NGS read is aligning from position 497 going all the way to 150 base pair. If it is 150 in total length, in this particular case, this is 37 nucleotides only. So this is shorter fragment. And then we get to see various different quality of alignment and all in the cigar trees and mapping quality, which is computationally interpreted by various different programs. Humans usually don't open FASTQ and SAMBAM files themselves. And after getting this alignment, we look at the variants, that means mutations in the sequencing dataset. So these variants are reported in the file format called VCF, variant call format. In this VCF file, again, we have headers at the top. Here, headers are identified by these hash symbols, and header again contains information related to the experiment. So, for example, this is following VCF 4.0 file format convention. Our reference is NGBI pilot 37 version, and then so and so various different other headers will be there. And then we'll have last line of header having column titles as per the variant reported, and each line of the VCF entry is one single variant. Again, this entire line has been broken down in this easy to use tabular format. But basically, what we need to pay attention to is that on chromosome number 2 at position 437, we have observed a mutation from G to A, and this mutation is associated with RSID RS6431. This RSID is very interesting for various different human genomics application. This is retrieved from DBSNP database. This DBSNP is a database of single nucleotide polymorphisms developed by NCBI, and this contains all the polymorphisms ever reported in human genomics. So this ID quickly allows us to understand whether the observed alteration is a potential germline alteration or not. Now, this is the first indicator. Just having RSID does not mean that this is a polymorphism in this particular patient case. So the second parameter that we can look at is allelic fraction. If you recall, a few slides ago, we saw how we calculated allelic fraction based on the count of altered base pair and count of the reference base pair at a particular location. And that is what we get to see in this VCF file. If allelic fraction is zero point 0.5 and if there is already annotated RSID, then one can much more confidently said that this is very likely to be a germline polymorphism. But again, this is not confirmation in the clinical settings or in the research setting. Usually, we take, other than the somatic tumor biopsy, we also take any of the other biopsies, say for example, blood, and we compare the mutations in the blood as well as tumor. If it is matching, then it is confirmed to be germline. If it is not matching, then it is confirmed to be somatic in nature. And somatic mutations can also have allelic fraction zero point 0.5. So please don't follow this number blindly, saying that zero point 0.5 is almost always a germline. No, that's not the case. Many of the somatic mutations are also observed at zero point 0.5 and 1 allelic fraction. Moving on, we have seen various different file formats that we typically use. Now, in this file formats, we report variants at various using various different convention, as well as in the report that we produce from these analysis from the VCF file. We follow various different convention. These conventions are represented as G dot, C dot, or P dot. G dot means the information provided is in the genomic scale. C dot indicates cDNA scale or a transcript scale, and P indicates protein scale. Now let's see some examples. In this particular sequence, you can see that in the reference genome there is A, and in the altered genome there is T. So there is a amino acid, a particular amino acid which is getting produced here has been mutated from K to E. So we can represent if this is chromosome 1, let's say, let's assume this is chromosome 1. So we can write CHR1 colon G dot 1, 2, 3, 4, 5 A to G. So on chromosome 1 at 1, 2, 3, 4, 5 position, there is A to G mutation. Okay. In the transcript level or protein level, we can write the transcript name or protein name. Let's say this is protein T53, then this is T53 P dot K123E. It is K to E mutation. Now, similarly, if we have a truncating mutation, then from A to T mutation, which is resulting into a stop codon, the premature termination, then we can write G dot 1, 2, 3, 4, 5 A to T, or a gene name or transcript name. If it is transcript name, then C dot. If it is protein name, then it is P dot. And here you can say K123 star. Star indicates a stop codon. Taking few more examples, here there is a synonymous mutation. You can see at the DNA level there is a mutation A to G, but it is coding the same amino acid. So at the DNA level, you will get to see that at 1, 2, 3, 4, 5 position there is A to G mutation happening. But when you do annotation using computational tools, you will find out that this is coding the same codon. So at protein level, there is no alteration. If you have say frame shift alteration, if there is an alteration happening here in which this particular nucleotide has been inserted, and after this insertion entire downstream sequence has been modified in such a way that this is resulting into uh, frame shift, then you can write that instead of A there is A to G, That means that there is G insertion has happened at the DNA level and which has resulted into protein level K to 1 to 3 E frame shift. So there is one nucleotide amino acid change happening as well from K to E, and then downstream sequence has been shifted. So we indicate as a FS frame shift. So, some real-world examples, just to quickly recall, EGFR exon 19 deletion is a very well-established therapeutic target which one get to see in cancer genomics very frequently, particularly in lung cancer and various other cancer types. At the genomic level, it will be CHR7 G, so and so position. This much nucleotide, this many nucleotides have been deleted, and the resulting only fourth nucleotide is remaining roughly about, not roughly, these are exactly 15 nucleotides which have been diluted, and that is generally referred to as exon 19 deletion in EGFR. In the cDNA level, it is C.2235, the codon position, deletion of 15 different codons in between these two. In the protein scale, P.E746 underscore A750 deletion. So deletion happening between 746 to 740, and which amino acids have been deleted? ELREA. Similarly, a substitution mutation, EGFR related habitat, another very common mutation, which is represented as CHR7 G dot so and so nucleotide position T2G, cDNA level, codon position 2573 T2G, protein scale P dot L85R. At the 85 amino acid position, L2R mutation happening. Okay, so now we have got some basics of how do we interpret cancer genomic channel. So now let's quickly see the pipeline that we use. So once we get the raw sequencing data, which is usually in the FASTQ format, we map it to the reference genome and we get SAM file. We remove marked duplicates and PCR duplicates, and we get resulting MAM files. Then we identify SNV and indel, and we get resulting VCF files. These VCF files are filtered and annotated by using standard quality filters, and then they are annotated with biological and clinical features. And at this stage, we start getting simple table which we can open in a Microsoft Excel or any other spreadsheet and generate visualizations and so on. So this is a more simplified version of a standard pipeline called VARSCAN BWA VARSCAN pipeline. There is another complicated version as well. My apologies, why it is yellow symbol. So in this more complicated version, we use BW GATK, and here what we do is we take a FASTQ file, map to the reference genome, remove the PCR duplicates, and then we perform two separate steps called realignment around indels and base recalibration. And then we go for HNV indel calling, and then similar workflow, filter, annotation, biological clinical analysis, and visualization. So these two steps have been specifically developed by GATK team, and hence this has resulted into a very widely and very popularly used pipeline called best practices BWGATK pipeline. So in this pipeline, let us see in detail what we do in these two steps. So, one may think, I mean, why are we getting into too much of complexity? And why can't we just simply align our NGS data to the reference genome and find out mutations like we get to see here? This gray indicates that there is no change with respect to the reference, and this base pair indicates there is a mutation. This is patient 1, 2, 3, and 4. Now, if you zoom out in this mutation area and if you see the nearby area, this is how this actually looks like. This is the actual patient sample that I have shown here, four different patient samples, all of them harboring this same mutation. But in the nearby region also, you get to see several different nucleotides, and all of the nucleotides indicate that there is a mutation happening in the nearby region as well. So now, out of these, all that we get to see, and all which we don't get to see in a particular visualization because human genome is pretty huge, which of these mutations are true? And identification of the true positive is a daunting task. It is like finding a needle in a haystack because of inherent nature of noise in the sequencing platform. Now, if you recall, the best quality score that we saw, Pret quality score a few slides ago, is 40. That indicates one wrong base pair out of every 10,000. Now, if you are sequencing 3 billion base pairs in the human genome, you expect several hundreds of false positives. So how do we identify a true mutation from this false positive? And that's where this all more complicated analysis is required. So the first one is to identify and mark PCR duplicates and remove them. So during the library preparation, whatever PCR step we are performing that accidentally overamplified some of the fragments. And if that fragment is also harboring mutation, it may look like there is a lot of mutation, but when we remove those duplicates, we learn that there is only single read representing that mutation, other reads are not representing that mutation. So now this is a very typical case where it can be a wrong base pair addition by sequencing of error. By error of polymerase that we use in the sequencing protocol. But if there are several different reads showing the same mutation, then we will be more confident to call it a true mutation. The second challenge that we get to see is that sequence alignment algorithm often produce this kind of artifact. In one of the NGS read, it will show that there is a alignment gap here, so there is a deletion happening here. In another read, it will show gap here, 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 and in one of the read, it is going much further away from here, from the actual location. So why this is happening is because of mathematical artifact. All the reads are 150 base pairs. So during the alignment, the best score of alignment is obtained in various different positions and various different settings. So for a aligner, it is very difficult to pick the very optimal alignment, and for the sake of speed, we have to compromise this accuracy a little bit. But then we learn to learn from the cancer genomics dataset that these results into artifact. These will basically identify three different deletions. One of these, and one of these. And we also tell that there is a missense mutation happening here. But once we do a more sensitive alignment, then it will align properly, and we'll get to see that there is a single deletion event happening here, and this particular variant was artifact. It is no longer present. So this is called indel realignment or local realignment around indel by using much more sensitive algorithm. But this is being done only in the known locations of indel. So we are not realigning the whole of the genome. So we are saving time in that way. But still, this is a very time consuming step overall. Then the last specific step that we perform is that GATK people figured out that Illumina platform has empirical biasness involved in the sequencing platform itself to give wrong quality score to the nucleotide. If this is the true reported nucleotide score, phase score, and this is the empirical quality score, then you get to see that this is not properly aligned together, and there is a systematic error happening. So this is corrected by GATK tool called GATK BQSR, and then it will be more properly, more appropriately aligned, and this will allow us to identify more accurately identify low quality scores and remove them from mutation analysis so that we can focus on the variants which are identified with higher confidence. Okay, so we have seen some complicated steps of genomics analysis, and they fit in a pipeline where we do alignment using BWA, then we do mark duplication by using Picard tools or some tools, then we use GTK for indel local realignment or to perform base quality score recalibration, and then we use again GTK for variant identification. Once we have a list of variants, we annotate them using a conventional database called DBSNP and Cosmic. These are freely available for academic purposes from their respective websites. Using these variant databases, we want to identify whether it is a potential germline or somatic alteration. So for a germline alteration, we have to have a paired normal sample, adjacent normal. If it is say lung cancer from left side of the lung, then we take another biopsy from the right side if feasible. If that is not feasible, then we retrieve blood of the patient and we compare whether a mutation is present only in the tumor tissue or also present in the normal. If it is also present in the normal, tissue, then it is a germline alteration. If it is absent in the normal tissue, then it is a somatic alteration. Sometimes we do not have paired normal, and in most of the tumor somatic settings, we usually don't have paired normal. So we use databases to identify which are potential somatic alteration. And one of the fantastic database is called Cosmic. database, and this cosmic database is a somatic database of all the somatic alterations in the cancer genome. So we use that to have a indirect annotation of a non-traumatic alteration. And then after doing all these annotation, we want to classify a mutation into either a benign or a pathogenic category. A pathogenic is the one which we'll use for targeting by using various drugs as per the standard guideline. And the benign are the ones which we will ignore and we will not target them. Sometimes a mutation is unclassified, and in that state, it is called variants of unknown significance (VUS), and this is what basic research laboratories focus into. They basically pick up these VUS and characterize these VUS by using cell lines and animal models and so on. So, now, in a typical cancer whole exome sequencing, one may get to see in the raw VCF file roughly about 30-40,000 different mutations. All of them are not true mutations. So if we apply 5x coverage filter, assuming that at least 5 different reads supporting a mutation indicate that this is a good quality filter, it is a good quality variant, then about 10,000 mutations will go away. Then if we remove all of the germline alterations, another 20,000 mutations will go away. If we look at the functional score, now this functional score means functional prediction by using computational tools. There are several tools called Polypen and Shift and Provian and various different tools are there. But there is also a database called DBNSFP, which has all of these functional tools compiled in the form of single database. So once you annotate your list of mutations using DBNSFP, you will get to see their functional score. Now, what this function score basically means is that based on the biochemical properties, based on the biophysical properties, based on the evolutionary properties, based on the nucleotide context properties, based on the protein location of the mutation, whether it is in kinase domain or any other functional domain of the protein, we try and predict whether a mutation is going to be deleterious to the protein function or not. So there are several different parameters involved in here, and we try and derive a mathematical score which can be numerically interpreted. And we shortlist all the 10,000 final mutations by using this functional score, and we find out that there are roughly about 100 mutations which are having enough of the functional score. One can easily assume that if median function score is 12 out of 30, then everything over and above 12 can be considered more functionally deleterious, and this will generally yield into very small number. And then we compare this set of mutations using biological and clinical knowledge, and we finally find out few handful of driver alterations for either clinical applications or for practice in the biological lab. So that was all about single nucleotide variants or a simple nucleotide variants, including single nucleotide variants and insertion deletion events. How do we analyze them? How do we find out them? How do we filter them? How do we annotate them? And what kind of concept we use to find out few handful of alterations to work with. Now let us quickly see another type of variant that we commonly find out in the practice, in the clinical practice is gene fusion. So for gene fusion, we use transcriptome sequencing. So we get started with RNA, we convert it into cDNA, chop them up, similar to what we do with the DNA. We ligate the adapters and perform the sequencing. From the sequencing, we want to find out gene fusion, and this can also be used for gene expression analysis, differential expression, and various other stuffs as well. But in the most clinical practice as of now is simple nucleotide variants and gene fusion. So let us focus more on the relevant part. So in this case, also we get raw data in the part Q, and this part Q is performed, with the quality checking by using a tool called part QC. Then we map it to the reference genome by using another aligner called STAR. This is specifically designed for RNA based alignment. In RNA, we know that there are several exons which are ligated together, and introns are spliced out during the central dogma processing. So this is not a typical alignment that we use in a DNA sequencing. Here we need splice aware aligner. So we don't use BWA as we use with DNA. Here rather we use STAR, and the STAR is a aligner which is familiar with all the splice junctions in the human genome. So this is splice aware alignment, but again it produces SAM-BAM files These SAMBAM files are processed for differential expression and then gene fusion by using a separate module of star aligner called star fusion. And then these fusions are annotated and visualized by using various programs. So in gene fusion, also there are some complexities involved, similar to what we saw in the DNA, where indel local realignment and base quality score recognition and so and so. In RNA-based fusion analysis or DNA-based fusion analysis, what we get to see is that 3' and 5' gene fusion can have untranslated region, it can have coding sequence, it can have untranslated regions, or it can have non-coding regions at both times. So it can be, in 2 versus 2 combination. The most common ones are the coding to coding sequence fusions, as those are functional. But you can see that out of a comprehensive cancer genome data set, one get to see all different kind of permutations in a real world data set. So this fusion analysis pipeline needs to be well aware, and all the fusion annotation tools need to be well aware of all different permutations. If you are using any particular tool which is having very good strength in say most common coding to coding analysis but not in other part, then you may miss out many of the well-known gene fusion. So you may have very high likelihood of false negative. So one needs to be very careful in choosing the correct bioinformatics pipeline for gene fusion. Now let us also see one real world example. EML4 ALK is a classical oncogenic fusion, most commonly reported in lung cancer and in many other solid tumors. So in this ALK is a kinase protein, and this kinase is typically required to be a driver in the fusion oncogene. And you get to see that in various different fusion transcripts, this kinase domain is very much intact. It is shifting a position little bit here and there, and similarly EML is also shifting position here and there, but in almost all of the cases you get to see kinase domain intact. In some of the rare cases you see kinase domain not preserved in the fusion product. So that is one of the parameter we typically use to assess the relevance of a fusion. And if the functional domain of the fusion is not preserved in the fusion product, then we don't consider it for biological and clinical applications. Another key parameter that we use in gene fusion is read coverage, similar to DNA sequencing. So here you get to see that fusion read is aligned. This is read 1 of gene A, and this is gene B. And this read is half mapping to gene A, half mapping to gene B. And that's it. There are several different reads which are, half mapping to both the sides of the gene. In a typical genome alignment view, you get to see that these reads are truncated. They are half mapping to this location one, and another truncated part of this read is mapping to a distant location in the genome. And if you make a coverage plot, this is how they look like. There is a high coverage in the upstream of the gene, and then coverage is dropping. And then in this upstream, there is low coverage, and in the downstream, there is high coverage. So upstream of this gene and downstream of this gene is shift together here. So this coverage and read count of the fusion junction becomes a very critical parameter to assess how well and how confidently are you identifying a gene fusion. So finally, to summarize everything, cancer genomics can deliver many pathogenic alterations and potentially useful alterations for therapeutic, diagnostic, and prognostic applications. Key parameters to look for in a cancer genomics report is sequencing coverage, variant allele fraction or VAP, which basically indicates whether mutation is heterozygous, homozygous, or very low VAP, which probably indicates false positive as well, a sequencing artifact. A target gene panel. If you are sequencing a particular gene panel having gene ABC, and then if you are saying that patient is negative for gene D, then that is a wrong interpretation. So always remember what you are sequencing and how you are interpreting. Pay attention to the genes which are excluded when you are doing a small panel based sequencing. You can't call them as a negative, and if you want to assess those other genes, then you need to again repeat your NGS experiment by using those genes in your sequencing assay. Then also pay attention to limit of detection, as in that at particular sequencing coverage, what is the VAP which can be confidently identified. Say, for example, we have seen in previous filtration strategy that 5x is a more typical cutoff that people use So in 100x sequencing coverage, 5x cutoff will basically allow you to identify variants of 5% and more VAP. If you find a variant with 1 to 5% VAP, then you know that this is probably a sequencing artifact that cannot be used for further follow-up without any additional validation. So that is your limit of detection for that particular ratio. With higher coverage, your limit of detection for VAP will keep going lower. And in a typical clinical setting, we usually do 250x or 500x coverage. So it allows us to find out VAP at much lower capacity, and you can further increase coverage to improve this as well. Then the last is to look at germline annotations, particularly to quickly distinguish germline and somatic. Here it is germline and somatic annotation both. Sorry, I missed out to write somatic here. So DBSNP and Cosmic are the two standard database which we use for a quick annotation of germline and somatic alterations in the cancer genome. Then we also pay attention to fusion read counts, particularly junction and spanning read pair counts, so that one can confidently interpret whether it is a reliable mutation, reliable gene fusion or not. Now, having said that, we also show some of the best practices of computational pipeline and one or two variants of them as well. One of the most commonly used best practice is BWA GATK pipeline, and for gene fusion, it is star fusion pipeline. But there are several other versions of this pipeline as well. So most of the genomic analysis tools are plug and play, means they use the very strict file format that we see FAQ, SAMBAM, VCF, and so and so. So these tools can be replaced by any other tool in a plug and play fashion. So if you are interested in trying out with your own pipeline with a unique combination of various different tools, which is not a part of best practice, feel free to do that. There is no restriction at all. But keep in mind that whenever you are doing these, you need to validate your pipeline, and this validation is absolutely essential for clinics as well as it is for biological research, because you definitely don't want to land up into some wrong combination of pipeline which is not taking care of some of the anomalies of the sequencing, which may result into various different false positives, and severely it can affect your clinical or biological outcome. So having said that, that's the end of my today's talk. Let me quickly tell you that this computational demand is increasing nowadays, and it is outnumbering YouTube, Twitter, and all the social media put together. Genomics data is growing about 10 times faster than even YouTube or Instagram or Twitter put together, and that requires a lot of computational resources. So recently we have established a high performance computing facility at ACTREC, which is a 6,000 square feet area facility. It has heterogeneous compute, which allows AI, artificial intelligence, genomics, proteomics, digital pathology, radiology, and so and so. And it has central storage pool of 2.5 petabytes, which is we are planning to increase this 1 petabyte per year. And this allows you to do scalable analysis. It has various different software solutions, and this takes the picture of our supercomputer. So if anyone is in need of supercomputer for academic purposes, please feel free to let us know. So with that, I'm coming to the last slide of acknowledgement. I'm thankful to Dr. Rajesh Sharin for giving me this opportunity to conduct this talk, and thank you to all the participants for patience. I'm also thankful to that lab and medical oncology with Dr. Omar Prabhat particularly for immense help in this initial data which I show for cancer genomics. All of my lab members and all the patients, all the collaborators who have helped me immensely. Thank you.
Okay. So one second. Hello. Hello.
Yes, sir.
Yeah. So thank you very much, Pratik. It was extensive. You covered everything, and I'm so happy that you started from very basic and explained everything. I think most people would have understood. And I'll ask just one question before I take up audience. You talked about that fusion you may miss, false negative. So what are the causes of a possible false positive fusion? That means there is no fusion, but your, as a bioinformatician, you think that there's a fusion and you don't have means for validation. Everything cannot be validated. So in what context or when do you suspect this may be a false positive fusion? You said few things, but if you can just elaborate, because I don't want people to get a false positive result.
Mhm. So false positive of gene fusion are common in Illumina-based sequencing, and that is because of chimeric reads spontaneously produced during the library preparation. It is a type of artifact happening mainly because of the probes that we used for capturing of the transcript. So these probes are often found to be, inducing various fusion reactions during the library preparation. Sometimes if we are using PCR based library preparation protocol, then also it can produce a gene fusion based on the wrong priming of the primers. So in such kind of cases, if fusion is occurring, then most likely we'll get to see very low read count. The coverage of that fusion junction will not be there. Spanning reads will be there, but the junction will definitely not be there.
Yeah.
So having 5x coverage of junction read is absolutely critical for clinical practice. In our own molecular tumor board, we use that cutoff. And
Yeah.
that's why I was highlighting in the summary that having junction read count and spanning read count is absolutely critical, even
Yeah,
for
you said
biological.
that. I just wanted you to emphasize that again because people may not understand that, and because everything you cannot validate it functionally or by other method. You
Yes,
have to just go by what
yes.
you get. Just one more general question I'll ask. You have seen sequencing technologies evolve over last 10-15 years. Previously, we used to had Roche 454, then Illumina, then again now many long read. So there some of the platforms had this problem like homopolymers or all those things issues. So current, the sequencing platforms that you see as a bioinformatician, okay, this we're using this Illumina or Roche. So what kind of things that you are, careful that you may be missing or getting a false positive because of the platform used?
So that's very interesting question. I haven't covered that because of time constraint.
Yeah. So, but I think for the audience to know that,
Yes,
that's why I
yes,
brought it up.
agree. So in human genome, we know that roughly about 30 to 40% of the genome is highly repetitive, and those repetitive regions are the same nucleotide or a repetitive patch of nucleotide repeated hundreds of times or sometimes thousands of times in a sequential manner. So it is very difficult to sequence them by using conventional sequencing platforms. When I say that, I mean first and second generation, ROS 454, Sanger, Illumina, and Torrent, all of them have difficulty in that. Third generation sequencing platforms, the long read sequencing platform, particularly Oxford Nanopore and PacBio, have some better capacity to handle short homopolymers, but even at a much longer length, they also start failing. So basically, this is one of the very difficult region, and analysis of them becomes very tricky. If one is doing typical cancer targeted panel sequencing, you don't need to worry because most of the target panels are not covering any of the homopolymer region. But if you are doing whole exome or whole genome, then it becomes very daunting task. So in bioinformatics, there are several tools which are specially designed for homopolymer realignment, similar
Okay.
to indel realignment. After getting the initial SAMBAM files, we use those realigners to get more and better accuracy of alignment in the difficult regions, and then we proceed with the analysis.
Okay. Thank you. So another question from Sriram Chandramurthy Pila. If the WAF is less than one, or means less than or D is less than one, is it always heterozygous or is it more complicated to identify a heterozygous? So homozygous should be 100% WAF. If it is say a 70% or 30%, then how do you infer?
So, very nice question. In genetic textbook, we see only zero point 0.5 and 1. But in practice, in cancer genome, that's not how we get to see it. So, cancer is a somatic disease, and all of the mutations we see in a tumor is usually not present in the whole of the tumor mass. It is present in very small fraction of tumor mass, and that results into VAP in the range of 0.001, typically infinite 0 all the way to 1. So everything which is below 1 is basically heterozygous. Everything which is 1 is purely homozygous. And when we say heterozygous, we refer to two different things here. One is clonal mutations and subclonal mutations. So when it is heterozygous, we want to find out in a typical tumor setting, we want to find out whether it is a clonal mutation, it is present in the whole of the tumor mass, or whether it is present in only a subset of the tumor mass. So everything which is, over and above zero point 0.5, in between zero point 0.5 to 1 is very likely to be a major clone and hence more likely to be a, clonal mutation, but it's not a black and
Yeah.
white kind of picture, so please take
Yeah.
it very carefully, but by and large it will be clonal in nature, and
Yeah.
everything which is below zero point 0.5 is more likely to be subclonal. That means it is
Yeah.
present in much
Yeah.
smaller fraction of the tumor.
Yeah. Thank you. You brought it out because a lot of people only think in terms of germline. In tumor depends what proportion of tumor is represented in the sample and what proportion of tumor cells have that mutation. So that is important. That you brought it out very nicely. And now Shreya Kashyap asked my no sorry Sindhura Gopinath, one of our uh, trainees, genetic counselor. Does the RSID change across different reference genomes? Like if the same data is aligned with GRCS 37 versus 38? A very
Mm.
basic question, maybe.
Yes, but very critical. I'm sure. I mean, people not from computational background are, definitely facing challenge in that. That's very nice question, I would say. So RSID would
Actually, I'll
never
just,
change.
sorry, 1 second. I'll just ask another question. You can cover both. Shreya Kashyap asked, my SNP was annotated as a promoter variant in DBSNP GRCS 37, but in 38, it's intronic. Since literature supports a promoter role, how should I treat it in functional analysis?
Yes. Okay. So we'll go through both. First, RSID will never change. RSID or cosmic ID is not with respect to reference genome, but it is a unique ID of a particular variant. So across various different reference genome, this can be compared together. However, if you have a VCF file and SAMBAM file obtained by a particular reference genome, then you can't directly compare it with another reference genome based file. For that, you will have to use a computational tool called lift over. You will have to lift everything from one version to another version and then compare them together. You will have to make the reference version. But if you are dealing with RSID or Cosmic ID, then you don't need to worry about reference
Okay.
version.
Okay.
Uh, the second question was about finding functional importance or functional relevance of a mutation if it is in a promoter region. So promoter-based mutations will require different kind of functional annotation tools, and fortunately, they are also present in the BNSFP database, the one which we discussed earlier. So here we will not be looking at the protein relevant values, but rather we'll be comparing the evolutionary parameters of a variant. Say, for example, a mutation has been at a particular site, a mutation has never been reported, and that site is functionally very much critically required for that change function. Then comparing 10, 20 different organisms will show conservation of that particular site. When a mutation in a cancer sample is very likely to be deleterious because it is essentially required as per the evolutionary evidence. The second parameter one can use is to map DNA regions against various protein binding sites, including transcription factors and so, which is also present as a annotation tool in the BNSFP. So if it is a part of any of the variant binding site, sorry, a protein binding site, then that mutation is very likely to be functionally deleterious. So you can use this kind of tools rather than protein-based tools.
Okay. Now, Fatma Islahi asked, how does the pipeline report novel variants in VCF files which are visible on IGV? Because variants that do not have RSIDs are not reported in DBSNP. So, how do you deal with this novel variants?
Correct. So novel variants will not have DBHNP ID as well as it will not have cosmic ID. So that's the way how we typically identify a potential novel variant. And this is the variant which we use for functional prediction. Because for the non-somatic alterations and other non-germline alterations, we usually know which are pathogenic and which are not. For a novel variant, we use functional tools as a first indicator of whether they are functionally deleterious or not. And second, we also look at their biological pathways and functions. In every experiment, whether it is a clinical investigation or whether it is a biological investigation, you will have a hypothesis, a premise in the mind. So based on that premise, which is the most potential pathways you are expecting to be altered, and if there are genes of those pathways showing alteration, then that's how you can narrow it down and focus further on those.
Okay, thank you. So you should know the biology, whether even for biological research also, you should know the question, what you are expecting. Now the
Absolutely.
Dr. Vada Party is asking how to interpret, asking multiple known genes come on report. So does higher the VAF means more significant? On a somatic, VAF is high, that means it is more significant.
No, WAP is not equivalent of significance.
Yeah.
WAP is only equivalent of significance when you are dealing with basic quality. If you
Yeah.
want to assess whether you are very close to limit of detection or you are in the safe zone of WAP for your sequencing setup, then it becomes indicator of significance. But other than that, WAP is not relevant to cancer related or therapeutics related significance.
Okay. So now very quickly again, Fatma Islai asks also for pipeline validation. What is, I suppose it is bioinformatic pipeline validation. What is the minimum patient data that should be sufficient for pipeline validation?
Well, typically, patient data is absolutely critical. Yes, I mean we work with we have seen in the field people use some 20, 50 patient data for various set of validations, in which previously they know what is the truth mutation set, but that is very difficult to get in a typical tumor sample because of a lot of heterogeneity. So what people do is they club a small set of patient data with a reference standard DNA and reference standard RNA. These are available commercially from various different companies, in which there is an absolute truth set of mutations induced by this synthesizing company, and you use that to benchmark your sequencing workflow, whether it is a wet lab based standardization or computational. It is suitable for both.
Okay. Now, very quickly, Shreya Kashyap will ask for the name of the databases. I think you have that. You go back and see the video. Many questions I want to finish. Rohit Sharma asks, can a person with no coding knowledge analyze data by themselves? And are there any automated platforms available for data analysis?
Yes. Another interesting question.
So
So,
everyone think now with the AI, you can do everything.
yes, yes. So, unfortunately, we don't have AI-based tools available yet, where you can, just like ChatGPT, you can ask the question that this is my path file, tell me which mutations are there. One day, perhaps it will be possible. Even my own lab is developing one such a tool called Can Insight. So once it is ready, maybe we'll talk about that. But even when it is ready, I would suggest that one should definitely learn the basics, fundamentals, and then use those tools. Whether you use an automated tool available commercially by a company, or whether you develop this pipeline yourself, or whether you collaborate with a computational laboratory and get it developed as per your custom need, in every single case, there is no alternative to learning fundamentals. Please be aware
Okay,
of that.
very important. So I think just two, three questions I'll take, then we'll finish. So Mohammad Sanawar Hussain has asked to avoid false positive results. It's immensely important to sequence normal tissue with tumor tissue. Otherwise, some research suggests that 28% false positive, but would be very expensive to do both. So, but this is important. So in real practice, we don't always do paired normal and tumor and normal this thing. So as a bioinformatician, how do you deal with it? Because you don't have the data from the normal, variant, germline variants, you don't have information. You mentioned in your talk, but again, if you can just summarize it.
Yes. So, in our own clinics, molecular tumor board, what we do is if there is a mutation in the tumor biopsy, and if it is having deviation P annotation, RSID, as well as if it is a part of genes which are commonly reported to be playing role in inherited tumors, then we ask for patient familial history of any cases. And if both of these are combined together, correlated, then we ask for germline confirmation of the mutation. Otherwise, typically we don't go for germline alteration straight away. Second thing that we use
Okay.
is we assess VAF in this particular case. if there is a mutation having VAF in, close to heterozygous range or close to, homozygous range, and then if there is any familial history in the patient family, then this is a very strong indicator that one should do germline testing and then probably consider for genetic counseling.
Okay, so Sheikh Gausinisa asked, want to know how to interpret data when there is a mismatch in sample and the reported, male or female. So you were told it's a,
Mm.
tumor tissue from this thing, and does it ever happen? I mean, I,
It rarely happens. So in the clinical settings, it usually doesn't happen
yeah,
because the whole workflow is much more robust and much more,
yeah, yeah,
standardized and much more tractable. So if it happens at every single stage, we get to know this way
yeah.
before in advance.
Yeah, it
But
could
in
be
research
pre-analytical.
settings,
You raise it to your clinician. You say, "This is what I'm getting." You said lung
yeah.
cancer from 60-year-old male. I can see. Now, Surya,
Okay.
so Anupriya Kaur says in exome, cancer exome data, in your experience, the type of capture kit used affects the genes or regions coverage. So the capture kit, would you like to answer this or?
Mhm. So there are several different capture kits available. For gene fusion, particularly, we use comprehensive gene panels, which are basically probe based, as they have more efficiency compared to the PCR based kits. But if you are doing only DNA based sequencing, then you can opt for either PCR based or probe based. They usually yield very similar results.
Okay, now the last question I'll ask Surya Mishra, how can false positives be minimized in liquid biopsy NGS given that cell-free DNA contains both tumor DNA and clonal hematopoiesis of chip of indeterminate potential related mutations? So in cell-free DNA, how can you distinguish between them? And I think I am stopping the questions now, they are still coming. Yeah.
Okay. Or maybe if there are unanswered questions, I will go to the chat box and I will put the answer over there if
Yeah.
anything is missed out. Now coming to this last question, cell-free DNA is something we haven't covered in the talk, but to basically summarize your question and the answer, cell-free DNA requires much more higher coverage. Usually we go with 5000x or 10,000x coverage, and that has another problem. Now, all of the DNA polymerase errors will also be piled up in the same location. So, distinguishing a low VAP somatic mutation from a DNA-based polymerase-based error is very difficult. So, we use something called error-corrected and barcoded sequencing library preparation for cell-free DNA, which makes it, which reduces error to the fold of 1,000-fold over and above the PRED score which can achieve. And the second thing that we do is we always try and correlate liquid biopsy with tissue biopsy as much as possible. If tissue biopsy is absolutely not possible, then one can definitely rely on liquid biopsy. In which particular case, one need to look at functional annotation tools and cosmic and DBHNP annotation retrieved so that one can find out whether it is a novel artifact or a novel mutation, potentially artifact, or a true somatic alteration.
Okay. So thank you very much. I think we'll end the session here. Pratik, you covered a very vast subject. You made it simple. And let me tell the people that AI is not going to replace an experienced, it is not just bioinformatics, it's a computational biology, that you have to correlate things. I mean, for some things maybe AI will get it right, but for many things it will not get it right, and because you don't have the background, you will not know where it is leading you towards the wrong side. So it is very important, make friends with a computational biologist, and those who do only hardcore bioinformatics, I would suggest please get into the biology part also, because then it makes a lot of sense. Because Pratik worked with a very top scientist, who was at ACTREC, now is in Delhi. So that plus working with clinicians gives you a different perspective. Your understanding is different when you attend lab meetings. There are people who are working with cell lines and all that. So I think that is very important. It is not just, so it is truly interdisciplinary. And Pratik is very collaborative. So if any one of you have any questions, you want to have a collaboration or ask questions, you can reach out to him in his first slide there was his email ID was there, and I would like to thank Pratik, and thank you very much. We have 3 more lectures in the series. After that, there are 12 seminars covering each of these sites. Thank you. Thank you.
Thank you.
We'll
Thank
end
you
the
so
meeting.
much, sir.
Okay. Bye-bye.
Okay.
Bye.
Goodbye, everyone.